Welcome to chapter 13, folks. This is all about the cytoskeleton. So this will develop your understanding from General Biology 1, when we talked about microtubules, microfilaments, and intermediate filaments. And our focus will be on microtubules and microfilaments. We'll only touch on intermediate filaments briefly at the end. So let's get going with just a quick overview, which will largely serve as review and will help to let me direct you to what you should kind of focus on as you are studying and thinking about different elements of the cytoskeleton. So this table here is really just reviewing the major structural elements of the cytoskeleton. Remember, those are microtubules, microfilaments, and intermediate filaments. And so as you look at this, it's going to be important to understand the basic structure here. So here is a microtubule. That's an assembly of protofilaments, which are assembled from alpha and beta tubulin heterodimers. So an alpha and a beta tubulin molecule associate to form a heterodimer, and then the heterodimers associate into chains to form protofilaments, and those protofilaments form these hollow tubule structures. Actin filaments are formed from the soluble G-actin protein, and those monomers polymerize, those proteins stick together and form these twisted fibers. And the intermediate filaments are more complicated in structure than either microtubules or microfilaments. They are formed from a twisted dimer of two independent intermediate filament proteins, and they then associate into groups of four, like you see here, and then those associate to make up the intermediate filament. So as you think about these structures, it's good to pay attention to the actual structure so you can compare the structures. Don't worry about these diameters. There's no use in knowing the diameters. It's important to understand what the monomers are, though. We'll talk about the 6 different classes of intermediate filaments. There's no real monomer there, but there are 6 types of intermediate filament we'll talk about towards the end of this section. The polarity of the microtubules and the microfilaments will become important, so appreciate that they have polarity, and we'll talk about that. There is no polarity in the intermediate filaments. Alpha and beta tubulin bind GTP or GDP, and actin binds ATP or ADP, whereas intermediate filaments bind neither of those nucleotides. And then you can think about the functions of the different cytoskeletal elements. I think this table is possibly a little bit more useful as it looks more at the relationship between structure and function. So sometimes this table will be a little bit more useful for you to look at the structure and the function relationships. And we will look at these arrangements that you're seeing down here using confocal immunofluorescence microscopy as we look at different types of intermediate filament, microtubule and microfilament, and look at their organization in cells. So the last thing to add in here is by way of dispelling a little bit of a myth. One of the things that people often raise as a difference between prokaryotes and eukaryotes is that prokaryotes lack any form of cytoskeleton, and that's not entirely true. There are structural features of bacteria which look very much like microtubules or look very much like other elements of the cytoskeleton. So, for example, there is a protein called FTSZ, and that is involved in cell division, and that is most like tubulin that forms into microtubules. And you can see down here there is some structural similarity between FTSZ that polymerizes into this polymer and the alpha beta tubulin heterodimers that polymerize into microtubules. So there's some structural analogy there between FTSZ and its polymer and alpha beta tubulin and its polymer. MREB is a protein that is most similar to actin, and that's often involved in DNA segregation and determining cell shape, and that's often what gives you this rod-like structure that you commonly see in organisms like E. coli. And then there's a particularly neat molecule called crescentin that you find in curved bacteria like Colobacter crescentis, and that's deposited in such a way as to cause growth in a curved manner. And so that protein regulates cell shape. So in prokaryotes, there are structures that are analogous to the cytoskeleton that you see in eukaryotes. So trying to dispel the myth that there's no cytoskeleton in the prokaryotes. Okay, that's it for our first section. As we move into microtubules and microfilaments, those sections are going to have multiple videos, and so I've broken those up into different parts just so they're not too long. I'll be back soon and we can start talking about microtubules. See you then.